So today, the work I'm going to tell you about is on behalf of a team that includes myself, but also Marcella Maas and Max Jan at MGH and Gabe Griffin at the Dana-Farber. So our interest and excitement about cell therapy is motivated by the fact that this is one of the approaches in oncology that's shown real long-term curative potential. And as these data in pediatric ALL indicate a 3-year survival rate of over 60%, it shows that when we get this therapy right, it can have truly transformative potential for patients. And the history of cell therapy is filled with heartwarming stories like that of Emily Whitehead, who was the first pediatric ALL patient treated with CD19 CAR T cells and is still cancer-free today. However, the picture in solid tumors is significantly different than the picture historically in cell therapy for heme malignancy. So this chart summarizes the overall response rate data for many key trials in this field. And on the left, you can see the average overall response rates for CD19 or BCMA-directed cell therapies for cancers like leukemias, lymphomas, and myelomas. Comparatively, the response rates for solid tumors, either for CAR T or TCR T cell therapies on the right, are much lower. And in reality, the cell therapy space has had significant challenges with solid tumors, where it's been very challenging to develop cell therapies with truly curative efficacy that makes itself apparent in early stage trials. And this has led some in the industry to believe that cell therapies will never work for solid tumors and to abandon their development. However, we don't share this belief, and we think that what we need to do is engineer newer and better cell therapies for solid tumors, and we're taking a multi-pronged approach to do that, starting from the first principles of T-cell immunology. So why are solid tumors such a challenge for cell therapy products? Well, the first and possibly the most important factor is that tumors have multiple mechanisms of immune evasion to inhibit and escape the effector functions of T cells. The best known of these is the PD-1, PD-L1 pathway, but this goes far beyond PD-L1. There are many, many layers of regulation that govern the effector functions of T cells, and tumors can use all of these to escape immune detection. And actually, these inhibitory signals can alter the differentiation trajectory of T cells, whereas T cells would normally differentiate from naive into effector T cells and then form long-term memory after the clearance of a viral infection. In solid tumors, the immunosuppressive signals and chronic exposure to high levels of antigen drive a different differentiation trajectory called exhaustion, where these cells are short-lived and hypofunctional. So we believe that if we're going to use T cells therapeutically, we need to take strategies, synthetic approaches, to alter this differentiation trajectory completely. We can use engineering techniques like gene knockout, overexpression, and the creation of synthetic response circuits to engineer new synthetic T cell states, states that may never have naturally evolved, but are indeed actually better suited for our intended therapeutic use for these therapies. So with that premise, we're taking a multi-pronged approach to the optimization of CAR T cells for solid tumors. First, we're making optimized CARs that are based on nanobodies that have improved signaling and antigen recognition capabilities, and we've shown that these are more effective than CARs based on scFvs. We're also taking multiple in vivo screening approaches, both loss of function and gain of function, to identify the gene edits that are best for optimizing the performance of CAR T cells, their expansion, persistence, and effector functions in solid tumors. And finally, we're developing drug-inducible control modules so that we can better control the efficacy and the safety of these CAR T cells using already FDA-approved drugs like lenalidomide. I'll touch on all of these different topics, but first I want to tell you about some of our exciting work doing in vivo screens to identify the edits that are best capable of improving CAR T cell therapy. So in collaboration with Marcella Maas's group, we've done the first in vivo CRISPR screens of CAR T cells in human tumor models using the approach outlined here. We take healthy human donor T cells, introduce CRISPR libraries with lentiviruses, and then transfer them into mice that have been engrafted with human cancers. We've done this for both solid and liquid tumors, and at the end of the experiment, we take the T cells out to figure out which ones have expanded the best or have the best effector functions. Using this approach, we've done screens to identify and rank the most important gene edits that you can make to T cells to improve their function in tumors. And in this particular case, we identified the cell cycle regulator CDKN1B as the top regulator of persistence in a BCMA-positive model of multiple myeloma. We then went on to show that the deletion of CDKN1B leads to curative efficacy in multiple myeloma models, several models of multiple myeloma, and all the mice are long-term survivors when we take this strategy. This is just one approach, and as I mentioned, we're taking several and we're combining them to optimize CAR T-cell performance in any context. So we're also generating optimized CARs using a new method that we developed to identify new single-domain antibodies or nanobodies using a cell-based immunization approach and nanobody-producing mice to create nanobody-based CARs that have better signaling and antigen recognition capabilities. And we've shown that these CARs significantly outperform SCFV-based CARs for treating solid tumors that express antigens like mesothelin. As I just mentioned, we're taking multiple screening approaches in vivo, both gain of function and loss of function, to identify the edits that are the best able to improve CAR T-cell function in solid tumors. And using this approach, we've identified both pan-model edits and edits that seem to be more specific to enhancing CAR T-cell efficacy in certain models, suggesting that the optimal genetic engineering strategy may be different for solid and liquid tumors, or different depending on tumor context. Finally, we're also developing new drug-induced control modules like this novel suicide switch that I'll describe here. So we've built this incredible suicide switch that gives us really rapid control over CAR T-cell lifespan that's based on caspase-activated DNase and its stoichiometric inhibitor, ICAD. What we do is deliver both CAD and a degron-tagged version of ICAD to T-cells at the same time, where the degron is activated to degrade ICAD only in the presence of lenalidomide. This then induces the degradation of ICAD and the activation of CAD to cleave the genome and initiate apoptosis. And we've shown that this can lead to the rapid and complete elimination of T cells upon exposure to lenalidomide. So where are we going with these approaches from here? Well, what we're really excited about now is that we're developing a new off-the-shelf allogeneic cell therapy platform that will enable the implementation of the technologies that I've just described. We're developing methods to enable the unlimited expansion of T cells in vitro, where they could be extensively engineered and then safely given to patients as an off-the-shelf therapy. This will result in an on-demand and low-cost cell therapy that has sophisticated engineering, the best edits to have best-in-class efficacy, and we're currently seeking partnership or investment to develop these CAR T cells using this new technique. So that's the end of my talk. I want to thank all the people from my lab, the Moss lab, Sen lab, and Jan lab at MGH, and the Griffin lab at the Dana-Farber for the amazing work they've done on this project. Thanks for your attention.